In my last video, I gave you an overview of the cloning experiment. Basically, what we're going to be doing in this experiment is doing a lateral transfer of the gene for GFP, green fluorescent protein, from this plasmid where it's currently found, pGFP, to a new plasmid called PQE31. And we talked about how we want to do that for purposes of putting PQE31 under the control of a promoter that allows us to control its expression. One thing I didn't mention before, but I will now, and we'll get into more details about it later, is that there's another advantage that PQE31 gives us when we put GFP into that plasmid. And that is that this PQE31 plasmid also contains something called a tag, a purification tag. And it's this thing right here, this 6X his. And essentially what this is a sequence that contains 6 codons for the amino acid histidine. And when GFP is put into this plasmid, the protein for GFP will be fused to these 6 histidine amino acids. And these 6 histidines act as what we call a purification tag or affinity tag that allows us to more easily purify GFP from bacteria, which is where the GFP will be expressed. And so this allows us to more easily purify this protein, and that's another reason why it's advantageous for us to put this gene GFP into this new plasmid. Now I want to give you an overview of how this is going to be accomplished. So if we look at two plasmids, any two plasmids really, where we want to do a lateral transfer of a gene, like GFP, there are a couple ways that this can occur. But in all the ways, what we end up having to do is open up the plasmid. It's circular right now. Ultimately, we have to open up that plasmid so that it can receive this gene, this piece of like GFP or any other gene that we're going to transfer over. If you think about a circular piece of DNA and a gene, which if we just took this gene out of this plasmid, it would be linear, right, in shape. How do we get something that's linear into something that's circular? Well, we're going to have to break open the circle at some place and open it up so that this linear piece of DNA can be inserted into it. And in fact, later on, we'll start calling this the insert, and we'll call this the vector because it's going to be receiving the insert. So we got to open up this plasmid DNA. The most common way to open this up is through a digestive enzyme called a restriction endonuclease, or a restriction enzyme. These are really useful tools in cloning and in molecular biology in that they allow us to cut double-stranded DNA and open up a circular plasmid so that it's linear. Restriction endonucleases are enzymes that are normally found, that were discovered in bacteria. Bacteria make a number of different restriction endonucleases. In fact, I think we're probably talking in the hundreds of different types of restriction endonucleases. And they all cut double-stranded DNA. So if you look at the ladder of double-stranded DNA, and here are the hydrogen bonds between the different bases, these restriction enzymes actually sit on top of the DNA, and they will cut the rails of the ladder. And they'll do so on both sides, so they actually have symmetric activity and can cut both sides of the DNA ladder. And what that ends up giving us is a break in the DNA strand. And so here this circular plasmid is double-stranded, and if we can find some restriction endonucleases, we can cut it open, and then it can receive this linear piece of DNA. Now, how does it receive it? Well, basically what you do is you mix these two pieces of DNA in a tube together with an enzyme called ligase and ATP. And ATP is another enzyme that basically does the reverse reaction of a restriction endonuclease. It actually seals the gap that was created in the rail of the ladder. And so if you add ligase and a linear piece of DNA and the vector together, you can actually create a new plasmid circular piece of DNA with the gene inserted into it. And this is a normal plasmid DNA. It would have all of the genes that it had before, plus this additional gene that we just added into it. Okay, so if we can cut the vector open, then it can receive this linear piece of DNA. But you might be asking at this point, well, this gene, like GFP, is in a circular piece of DNA too, so how are we going to get it out of that plasmid so that it's a linear piece, so that we can insert it over here into this vector? Well, the same thing. If we can if we can cut right here and right here with restriction enzymes, then that will release this linear piece of DNA. Of course, we'll have another piece of DNA left over, which would be the rest of the plasmid, but we can actually separate this linear piece, which will be our insert, from this original host vector, and then we can take this insert and combine it with this new vector and add ligase and seal it so that it's a circular plasmid again. So if we have places right here and here where the restriction enzymes can cut, and we have a place here where the restriction enzymes can cut, then we can cut this vector open, and we can cut out this gene, and we can ligate them together. Okay, so let's start adding some complication to this, or let's add some complexity, I should say. Hopefully not complication, but complexity. So the restriction endonucleases do cut double-stranded DNA, but they don't cut it just any old place. They are very specific, depending on which restriction endonuclease we're talking about. They're very specific for particular sequences of DNA that they recognize and bind to and cut at. And so not every part of the DNA can be cut. What this means is, if we want to cut this open with restriction endonucleases, there needs to be somewhere on this piece of DNA what we call a restriction site. And this would be a particular DNA sequence that the restriction endonuclease recognizes. These restriction sites typically are about 4 to 6 bases long, and like I said, there are probably in the hundreds of different restriction endonucleases, so there are probably, for each one of those, there is a unique restriction site or a unique sequence that it recognizes. By and large, these sequences, these restriction sites, are palindromic, meaning that the sequence reads the same in both directions on the DNA strand. So if we have our double-stranded DNA here, and I had a sequence that's AAG up here, then my sequence would read AAG down here, and then of course this sequence across from it would be CTT and CTT. And so if we looked at this sequence and read it on the top strand here, it would be AAG CTT, and on the bottom strand it would be AAG CTT. This is called a palindrome. Most restriction sites are palindromic. All right, so if we want to cut this vector open, we need to have a palindromic restriction site here. Typically, vectors that are produced for people to clone will actually artificially engineer multiple palindromic restriction sites all into one area, so that you can have flexibility in which site you choose to cut open the DNA. And I'll tell you why that would be important here in a minute. But when you see a bunch of restriction sites that are all kind of clumped together to allow you to cut out the piece of DNA, or sorry, cut open the plasmid here, We call this a multiple cloning site, or MCS. If we look back at the plasmids that we're using for this project, we can actually see that they have multiple cloning sites. So PGFP actually has a 5' multiple cloning site and a 3' multiple cloning site, and the PQE31 also has a multiple cloning site. In this map, they've actually listed for us all of the sites that this multiple cloning site has. So it has 1, 2, 3, 4, 5, 6, 7, 8, 9 different sites, 9 different palindromic sequences from which we could choose to cut one of them and open up this PQE31 to make it linear so that our PGFP could be inserted inside. So this is good news. This means that there are locations where we can cut out the GFP gene, and there are locations where we can cut to open up the PQE31 plasmid to receive the GFP gene. Okay, let's add a little more complexity to this. As I told you, each of these sites is a different palindromic sequence, and if we want to fuse two pieces of DNA together, it's important that those two pieces of DNA were cut with the same enzyme, so that they were cut at the same palindromic sequence. So let's just take, for example, this enzyme here, this restriction enzyme site called KPN1. And I can actually look on the internet to find what actual sequence it cuts. Okay, I went to a site called New England Biolabs and found the sequence for KPN1. So this is a particular enzyme, and the site that it cuts at is GGT-ACC, read in the 5' to 3' direction, so that on the other strand, which is anti-parallel in the 5' to 3' direction, we also would read GGT-ACC. So see how it's palindromic? They also indicate where in the DNA ladder, where on the rails of the DNA ladder, so the phosphodiester bonds, this enzyme cuts. So it cuts between the two C's. So that PQE31 plasmid has a KPN1 site in it. So what would happen if I mix this plasmid with KPN1? It would cut right here between the two C's, as we saw on that previous slide. And so what would we get after we cut that? Well, we'd get a linear piece of DNA that looked like this. So I'm drawing it in a circular shape, but I want you to realize that it's not a full circle anymore, because I've cut right through here, and so now it's really just a linear piece of DNA, but I kind of wanted to show you, its former circular shape. And what I want you to notice is that now there are some unpaired bases that are part of this piece of DNA. Right here, 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 and here, and here, 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 and here. The GTA and C and the GTA and C over here. These unpaired bases are called sticky ends, and the reason they're called sticky ends is because they have a lot of affinity for their match, for their pair. So C has an affinity for G, A for T, T for A, and G for C. And so if we can find another piece of DNA that has that same sequence, they will actually stick to each other fairly well. Now you'll probably notice that over here we have the exact match that these guys are looking for. There's a C, an A, and T, and a G, which would love to anneal with the C, A, T, and G here. And if we put this plasmid alone in a solution, they would anneal to each other. These two groups of 4 unpaired bases would anneal to each other. But it wouldn't become a circular piece of DNA at that point unless we added ligase, which would then reseal that piece into a circular plasmid. They would just be annealed to each other, but they'd be loosely held. And ligase then comes in and would actually recircularize the plasmid. Remember, in this project, what we want to do is we want to take GFP from this other plasmid, PGFP, and we want to cut the GFP out right here and here so that it's free, and then we want to insert it into here. Well, how are we going to do that? Well, we need to make sure that when we cut it at both ends with KPN1. Because if we do, then the ends on the end of GFP will be compatible with the ends here on this plasmid. So we'd have a G, T, A, and C on this end, and we'd have a G, T, A, and C on this end, and this would be our GFP gene here in the middle. And so if we had this kind of sticky end on the GFP gene that matches the sticky ends that we find up here, then this GFP will stick well or anneal well into this plasmid. And so, in order for this to work, we would have to have a KPN1 site on both sides of GFP so that we would get a sticky end on both ends of GFP that match the sticky ends that we see on the PQE31. Okay, so if we did this, if there indeed were KPN1 sites on both sides of GFP and the KPN1 site here in PQE31, then we could do this cloning, and it would be called cloning with via restriction endonucleases. I want you to recognize something, though, before we move on, and that is I want you to look at these two ends, these sticky ends that we've created by cutting GFP with KPN1 on both sides. I want you to ask yourself, where would these ends be compatible up here on the PQE31? Well, we have a C, A, T, and G, which would be compatible here, wouldn't it? The C could go there, the A could go there, the T could go there, and the G could go there. But I also want you to see this could fit nicely over here, too. The C could go there, the A could go there, the T could go there, and the G could go there. And this C, A, T, G could fit nicely into here. And if we did that, then we'd end up putting the GFP in backwards. Now, you might say, well, would that work? No, it would not, because this, if we look at this plasmid, the PQE31, it is reading the DNA in this direction. And if we put the GFP in here at this point backwards, then the DNA won't be read correctly, and this gene will be read backwards, and it'll end up producing nonsense. Probably it won't even produce a protein. It'll probably produce some shortened protein that's not even closely related to GFP, because we're reading it backwards. It'd be like reading a book backwards. It's going to be gibberish. I want you to recognize that by using just one restriction enzyme, KPN1, we have the risk of putting that GFP gene in backwards. And in fact, in 50% of your ligations, when you add ligase, the gene will go in backwards. So one way we can avoid that is by cutting with two different restriction enzymes. And that's where the idea of having multiple cloning sites comes in handy. Because if we cut with two restriction sites, like KPN1 and SAL1, for example, then if we look at what that would create, I'll go to a new slide here. So here we have our PQE31, and there's a KPN1 site here, and a SAL1 site right next to it. If I add both enzymes, both KPN1 and SAL1, I will get a linear piece of DNA after the cut with this tiny in-between fragment here that will kind of get lost in the mix. but we don't want to forget that it would exist. And one end would be compatible with KPN1 ends, and the other end would have a Sal1 sticky end. So these two ends here would be sticky ends, but they would be different sticky ends. So they actually wouldn't have affinity for each other. They would have affinity for these ends up here, but this piece we're going to get rid of through a purification process. And so actually, now these two sticky ends are not compatible with each other. So even if I added ligase, they wouldn't come together and reseal the circle. Now, if on my PGFP plasmid, here's my GFP gene from here to here. If I have a KPN1 site right here and a Sal1 site right here, then I can cut out this GFP gene with both of these enzymes, so KPN1 and Sal1, and I can get this linear piece of DNA that has my GFP gene, and here would be, this end would be compatible with KPN1 sticky ends, and this end would be compatible with Sal1 sticky ends. Now when I mix these two things together, my vector and my insert, they will actually, this end would anneal to this end, and this end would anneal to this end, and when I added ligase, I would get a circular piece of DNA religated that now has my GFP gene in the middle. So we'd call this PQE31 / GFP because now it's still PQE31 but now has GFP added into it. So by using two different restriction enzymes, I can do something called directional cloning, which basically means that I have forced 100% of my inserts to go in the correct direction. They won't be able to go in backwards because the KPN1 site is not compatible with the Sal1 site, and vice versa. So this is typically the way it's done. You typically would cut with two different enzymes rather than one. Okay, let's add a little more complexity to this problem now. What if you took a look at the maps of PGFP and PQE31 and there weren't compatible restriction sites? So we call this KPN1 site compatible with another KPN1 site, and we call the Sal1 site, we call it compatible with this Sal1 site, this Sal1 sticky end. What if there weren't compatible sites? So what if the KPN1 site didn't exist here and the Sal1 site didn't exist there? In fact, let's take it to the extreme case. What if there were no restriction sites anywhere on either side of GFP? What could you do in that situation? Okay, before I go on with that, I just realized I need to go back and say one more thing about restriction enzymes and the sites. When you decide to cut with these two different restriction enzymes, if you can, and if indeed that's possible, do make sure that you examine your gene itself and confirm that it does not contain either of these two restriction sites. Those restriction sites must be flanking the gene. In other words, they must be outside of the coding sequence. So if this is the start codon and this is the stop codon, these cut sites must be outside of that coding sequence, which would be right here between the start and stop codon. If you had a KPN1 site right in the middle of the GFP gene, I hope you realize that if I add KPN1, it's going to cut in three different places, here, here, and here, and I will get two fragments of GFP. And while you might be able to ligate that into your PQE31, chances are it's going to greatly decrease your efficiency and your ability to get a product that has the intact GFP gene in it. So you always have to make sure that the inside of the gene does not contain any of the sites that you're planning on using to cut with. Okay, now getting back to this idea of what happens if there aren't any restriction sites here. Let's say that there is a KPN1 site here and there is a SAL1 site here, but there aren't any restriction sites here. This plasmid was poorly made for whatever reason, and just, or just not necessarily poorly made, but just by, there's lots of different plasmids people make, and they make them for different reasons, and sometimes they're just not going to include the restriction sites that would be most useful for you. And so there aren't the restriction sites that you need on either side of GFP, flanking it. What can you do? So there is another option for cloning this GFP gene. You're not out of luck. You still can do it. But what you're going to have to do is take a different tactic and do something called PCR cloning. Okay, PCR stands for polymerase chain reaction, and we'll have another video on how PCR works a little bit later. But what we're going to do with PCR cloning is, instead of cutting out, so here's our GFP gene in pGFP, instead of cutting this out from the plasmid, instead I'm going to make a photocopy of it. Okay, not literally a photocopy, but I'm going to make a molecular copy of it through a process called replication. Now, you guys are very familiar with that process. Replication is when DNA replicates, when the cell divides, right? I'm going to use an artificial replication that I'm going to do in a tube, so it's basically in vitro, in a tube, in vitro replication. So via this in vitro replication, I'm going to make billions of copies of my GFP gene. And in the process of that replication, I'm actually going to add a little piece onto both ends of my GFP gene that was not in the pGFP plasmid. So these are additional sequences that I'm going to add to either end of the GFP gene. And what are these sequences going to be? Well, maybe you've guessed by now, they're going to be restriction enzyme sites. And they're going to be restriction enzyme sites that are compatible with the restriction sites that I'm going to use to cut the PQE31 open with. So on this back side of GFP, at the end of GFP, I'm going to put SAL1 sites. And on the front side of GFP, I'm going to put KPN1 sites. Now, I can only add those sequences if I do PCR. So PCR is a way to copy the gene through replication, but in that process I can actually add some sequences. Basically how this works is, when you do PCR, you have, like you do in DNA replication in the cell, you have a primer that will anneal to the DNA just upstream of the GFP, and a second primer that will anneal just downstream of the GFP gene. And these primers will anneal to the DNA and indicate where the polymerase should copy. Remember, when we do replication, the enzyme that's doing the replicating or the copying of the DNA is called DNA polymerase. So we're going to add a DNA polymerase here, and it will attach right at the end of the primers, and it will copy what's in between the primers. Now what ends up happening is that the primers end up being included in that copy. And so if I have a primer here, and the DNA polymerase copies the DNA in between, I'll actually end up with a piece of DNA that contains the gene and the two primers on either end. The primers become part of the PCR product. And I'll have another video where I show that a little bit better. But I can artificially I can synthesize primers that also have an extra sequence that does not match with the pGFP gene, that has a KPN1 restriction site on one end and a SAL1 restriction site on the other end. And if I add those sequences to my primers, then the PCR product I get will contain, in the end, a KPN1 site and a SAL1 site. Now that I've done that, and I've made copies of GFP and it contains those restriction sites, now I can do restriction enzyme cloning with directionality, right? Because now I have two different sites on each end. And so I can take these copies and I can cut them all with KPN1 and SAL1, and I will get a bunch of GFP copies that have compatible sticky ends that are compatible with the sticky ends that I've produced on PQE31. And so my problem is solved. I don't have to worry about the fact that there weren't any restriction sites there. I can just make them in the process of PCR. So this process of PCR cloning is very commonly used in order to overcome the obstacle of not having compatible restriction sites.